Aging, the gradual and inevitable process of physical change that occurs in every living organism, has long been considered an immutable fact of life. It is marked by the progressive decline in physiological functions necessary for survival and fertility. Over the years, science has unraveled many mysteries about aging, shedding light on why our cells and tissues degrade over time. But what if we could not only slow down aging, but actually reverse it? What if the decline that comes with aging could be prevented, treated, or even cured? Recent advances in biotechnology have opened unprecedented possibilities. Scientists are now exploring and developing various technologies that target the root causes of aging. These technologies are not merely interventions to extend lifespan, but revolutionary methods aimed at enhancing health span, the period of life spent in good health. This new era in medicine is not just about adding more years to life, but more life to those years, allowing us to live healthier longer. Aging can be understood through several types of cellular and molecular damage, each contributing to the aging process in distinct ways. 1. Cell loss and atrophy, where cells die off faster than they can be replaced, leading to weakened tissues and organs. 2. Nuclear DNA mutations, accumulations of genetic mutations that can cause cancer and cellular dysfunction. 3. Mitochondrial DNA mutations, defects in the mitochondria, the power plants of our cells, leading to energy deficits and cellular damage. 4. Intracellular aggregate, the buildup of unwanted proteins inside cells that disrupts their function. 5. Extracellular aggregate, similar protein buildups outside cells, which can interfere with tissue function and health. 6. Cellular senescence, aging cells that refuse to die, secreting harmful substances that lead to inflammation and tissue deterioration. 7. Extracellular matrix stiffening, hardening of the tissue scaffolds in our body, which affects the functionality and elasticity of tissues. 8. Dysregulated epigenetics, changes in how genes are expressed without altering the DNA sequence, affecting cellular function and longevity. Each type of damage has sparked the development of specific technologies aimed at repair and rejuvenation. 1. Stem cell therapy and regenerative medicine replenish lost or dysfunctional cells, restoring tissue integrity and function. 2. Gene editing techniques, such as CRISPR, allow precise corrections in DNA to prevent or reverse mutations that lead to disease. 3. Senolytics specifically target and remove senescent cells, reducing inflammation and promoting healthier tissue environments. 4. Anti-amyloid therapies and immunotherapy clear harmful protein aggregates that are hallmarks of diseases like Alzheimer's. 5. Advanced glycation. End product breakers in tissue engineering are developing ways to maintain and restore the elasticity of tissues, crucial for preventing organ decline and improving vascular health. 6. Epigenetic therapy uses drugs or gene modification to reset gene expression to youthful levels, potentially reversing the effects of aging at a fundamental level. These technologies are not mere science fiction. They are part of an emerging arsenal in the fight against aging. With each breakthrough, we edge closer to a world where aging can be controlled and perhaps cured. This is a world where each advance not only promises longer life but offers a profound improvement in the quality of life. The pursuit of these technologies is not just a pursuit of longevity, but a pursuit of a healthier, more vibrant existence. As we stand on the brink of these transformative advancements, the possibility of turning the tide against aging has never been more within reach. As we age, one of the primary challenges our bodies face is the phenomenon known as cell loss and atrophy. This occurs when cells within our tissues and organs gradually die and are not replaced quickly enough, if at all. Over time, this cell loss leads to diminished function in the affected tissues, contributing significantly to the decline we associate with aging. For example, in the brain, loss of neurons can lead to cognitive decline, while in muscles, it results in weakness and reduced mobility. However, imagine a future where aging is not an inevitable descent into decline, but a challenge to be mitigated through advanced technology. This future is being built today through the fascinating field of regenerative medicine. Two of the most groundbreaking technologies in this arena are stem cell therapy and tissue engineering, both of which have the potential to reverse cell loss and atrophy and restore youthful functionality to aging tissues. Stem cells are the body's raw materials, cells from which all other cells with specialized functions are generated. In stem cell therapy, these powerful cells are employed to repair or replace damaged tissues and organs. The process often involves harvesting stem cells that have the capacity to differentiate into various types of cells, then treating these cells in a lab to multiply and specialize according to the needs of the body part they are meant to heal. For instance, scientists can now grow stem cells that will become heart muscle cells, brain cells, or any other type of cell. These can be reintroduced into areas of the body where cell loss has occurred, offering a reset button for aged tissues. Imagine a heart weakened by age being rejuvenated, its worn cells replaced with new, vibrant, beating cells, all thanks to stem cell technology. Tissue engineering takes this concept even further by combining these versatile stem cells with scaffolds, materials that act as templates for tissue formation. These scaffolds guide the stem cells in forming structured functional tissues. Engineers can design these scaffolds to mimic the complex structures of natural tissues, from the spongy texture of bone to the intricate networks of blood vessels. In practical terms, this means that scientists can actually construct organs in the lab. For patients with liver or kidney failure, this could be a game changer, offering them bioengineered organs free from the risks of rejection and the wait lists associated with traditional transplants. The implications of these technologies are staggering. As they evolve, we could see them not only being used to treat individual cases of disease or injury, but also as preventive measures against the degradation of age. Imagine a world where every decade of your life could be greeted with a cellular tune-up, keeping your tissues as functional as they were in your youth. We are standing on the brink of a revolution in medical science. The technologies being developed today in regenerative medicine could extend our healthy lifespans, dramatically reduce the burden of age-related diseases, and improve the quality of life for millions around the globe. With ongoing research and support, the potential to heal ourselves from within, cell by cell, is within our grasp. This isn't just a promise, it's the bright dawn of a new era in human health and longevity. Our DNA is the blueprint of life, encoding the instructions that guide the growth, development, and functioning of all living organisms. In humans, this genetic material is housed within the nucleus of every cell, orchestrating the complex symphony of life processes. However, as we age, mutations, changes to the DNA sequence, can accumulate. These mutations can disrupt how cells function, leading to diseases such as cancer and contributing to the aging process itself. Nuclear DNA mutations can be caused by a variety of factors, including environmental toxins, radiation, and errors during cell division. Some mutations may be harmless, but others can lead to serious consequences. For instance, a mutation in a gene that controls cell division can cause cells to divide uncontrollably, a hallmark of cancer. But what if we could correct these mutations as easily as correcting typos in a document? Enter the cutting-edge fields of gene therapy and genome editing, technologies poised to transform our approach to medicine by fixing these genetic typos at their source. Gene therapy involves introducing a correct copy of a gene into a patient's cells, compensating for the defective or mutated version. This is typically achieved using vectors, often viruses that have been genetically engineered to be safe, which carry the correct gene copy into the cells. Once inside, this new gene can produce the right protein, restoring normal function to the cells. Imagine a world where genetic diseases like cystic fibrosis or hemophilia could be permanently cured with a single treatment, restoring health and vitality to millions. Taking this a step further, technologies like CRISPR-Cas9, a revolutionary genome editing tool, allow scientists to cut DNA precisely at a specific location. This enables them not only to remove mutated sections, but also to replace them with correct sequences. It's like using a molecular scalpel and glue, cutting out the defective parts and pasting in the right ones. The potential applications of genome editing are vast, from treating inherited diseases to potentially enhancing our DNA to better resist diseases of aging, such as Alzheimer's. This technology isn't just about fixing errors. It's about potentially upgrading the human genome in ways that could extend life and enhance health dramatically. These technologies are developing rapidly, with new breakthroughs reported regularly. Clinical trials are underway for various genetic diseases, and each success brings us closer to a future where DNA mutations are no longer a sentence to disease or decline, but a problem that can be fixed with precise, targeted interventions. The excitement surrounding gene therapy and genome editing is well-deserved. We are on the cusp of a new era in medicine, where our very genes can be edited with the precision of computer code. This isn't just about treating diseases, it's about redefining the boundaries of human health and longevity. The future where genetic disorders are a thing of the past is within our reach, promising a healthier, longer life for all. With these technologies, the next generation might look back on many medical conditions as we look back on the diseases of the past that have been eradicated by vaccines and modern treatments. Welcome to the forefront of genetic revolution. Imagine the mitochondria as the powerhouses of our cells, generating the energy needed for almost every cellular activity. These tiny structures within our cells have their own DNA, mtDNA, distinct from the DNA in the nucleus. However, mitochondrial DNA is particularly vulnerable to damage due to its close proximity to the energy production sites, which generate harmful byproducts like reactive oxygen species. Mitochondrial DNA mutations can disrupt the normal functioning of cells, leading to reduced energy production and increased oxidative stress. This can contribute to a range of diseases, particularly those affecting high-energy demand organs like the brain, heart, and muscles, and is also implicated in the aging process itself. Diseases linked to mitochondrial dysfunction include neurodegenerative disorders like Parkinson's and Alzheimer's, certain forms of muscular dystrophy, and even diabetes. What if we could repair these fundamental energy generators and restore them to their youthful efficiency? Recent advances in biomedical technology are turning this possibility into reality through groundbreaking approaches like mitochondrial replacement therapy and mitochondrial gene editing. Mitochondrial replacement therapy, MRT, is a novel technique initially developed to prevent the transmission of mitochondrial diseases from mother to child. This procedure involves replacing unhealthy mitochondria in an egg or embryo with healthy ones from a donor, ensuring that the offspring are free of mitochondrial disorders. This method not only prevents disease but also holds the potential to rejuvenate cells by replacing aged, dysfunctional mitochondria with fresh, fully functional ones. On a more microscopic level, new technologies are being developed to edit mitochondrial DNA directly, offering a way to repair mutations without replacing the entire organelle. Techniques like CRISPR-Cas9, traditionally used for editing nuclear DNA, are being adapted to target mitochondrial DNA with precision. Researchers are exploring systems like CRISPR-Cas9-derived mitochondrial base editors that can specifically alter the mitochondrial genome, correcting harmful mutations directly at their source. Imagine a future where mitochondrial diseases are a thing of the past, and where the natural aging process is significantly slowed thanks to restored mitochondrial function. The implications are profound: healthier aging, delayed onset of chronic diseases, and enhanced vitality through decades. The excitement around these technologies is palpable in the scientific community, as they hold the promise not just to treat, but potentially to reverse the effects of aging and disease at the cellular level. As research progresses, the potential to apply these techniques more broadly could transform our approach to a wide array of diseases that currently have no cure. As we stand on the brink of these incredible scientific advancements, the possibility of controlling our cellular health and energy production through mitochondrial therapy is not just science fiction, it's becoming science fact. This burgeoning field of mitochondrial medicine could be the key to unlocking prolonged, healthier lifespans, heralding a new era of preventive and therapeutic options that target diseases at their very source, the cellular power plants of our bodies. Welcome to the exciting frontier of mitochondrial rejuvenation. In the bustling cities of our cells, Various proteins perform countless tasks, each critical to our health and vitality. However, sometimes proteins misfold or clump together, forming what are known as intracellular aggregates. These protein clumps can disrupt cellular functions, leading to cellular traffic jams and breakdowns that impede the cell's ability to operate efficiently. Over time, this accumulation can contribute to diseases like Alzheimer's, Parkinson's, and Huntington's, where aggregates become toxic to cells. Intracellular aggregates interfere with cell functions by occupying space and hindering processes essential for cellular survival and communication. They can also induce stress responses in cells that lead to premature cell death. The presence of these aggregates is not just a sign of disease, but often a driving force behind the progressive nature of neurodegenerative disorders. Imagine if we could send in a cleanup crew specifically designed to clear this molecular debris, restoring cell function and preventing disease progression. This is not just a dream. It's becoming a reality through innovative approaches like enhanced autophagy, proteolysis targeting chimeras, PROTACs, and molecular chaperones. Autophagy is the cell's natural process of cleaning out damaged or unnecessary components, including protein aggregates. By enhancing this process through drugs or genetic modification, scientists aim to boost the cell's ability to clear out these harmful aggregates more efficiently. This approach is like upgrading a city's waste management system to be faster and more efficient, ensuring that cellular pathways remain clear and functional. Proteolysis targeting chimeras, or PROTACs, represent a cutting-edge development in protein degradation technology. PROTACs are designed molecules that can tag specific, unwanted proteins for destruction by the cell's waste disposal system, the proteasome. This method is highly selective, targeting only harmful or misfolded proteins while leaving healthy ones intact. It's akin to having a smart robot that can sift through a pile of refuse, picking out only the toxic materials and leaving the rest untouched. Molecular chaperones are another fascinating solution. These naturally occurring molecules help proteins fold into the correct shapes and can refold misfolded proteins or prevent them from aggregating. Enhancing the production or activity of these chaperones through genetic therapy or drugs could help maintain protein health and prevent the formation of harmful aggregates. The technologies to tackle intracellular aggregates are evolving rapidly, with potential applications that could revolutionize the treatment of neurodegenerative and other protein-related diseases. Clinical trials and research are expanding our understanding and capabilities, bringing us closer to a future where diseases caused by protein aggregation are manageable or even reversible. Imagine a world where Alzheimer's or Parkinson's are no longer life sentences but treatable conditions, where the aging brain retains its clarity thanks to cellular janitors, empowered by the latest technology. This is the world that the groundbreaking fields of enhanced autophagy, PROTACs, and molecular chaperones are aiming to create. The promise held by these technologies is not just in treating symptoms, but in addressing the root causes of diseases, potentially restoring full cellular function and transforming the landscape of medical treatment. The journey to decode and declutter our cellular inner workings is well underway, heralding a new era of health and longevity. Imagine our body as a bustling metropolis where roads and pathways must remain clear for traffic to flow smoothly. In this analogy, extracellular aggregates are like debris that blocks these pathways, interfering with essential communications and functions between cells. Extracellular aggregates consist of proteins and other substances that accumulate between cells, disrupting tissue structure and function. This kind of damage is a hallmark of diseases like Alzheimer's, where amyloid beta plaques form in the brain, and in systemic amyloidosis, where protein fibrils deposit in various organs. As these protein plaques build up, they can impair nerve cell communication, leading to memory loss, cognitive decline, and other severe consequences. The presence of these aggregates can also provoke inflammation, further damaging surrounding tissues and exacerbating conditions such as heart disease and diabetes. What if we could clear these obstructions effortlessly, restoring the fluid communication between cells and revitalizing tissue function? Modern medicine is on the brink of making such capabilities a reality through exciting technologies like immunotherapy and enzyme therapy. Immunotherapy leverages the body's immune system to target and clear extracellular aggregates. This approach involves creating specialized antibodies that recognize and bind to these protein plaques. Once bound, these antibodies flag the aggregates for destruction by the immune system's cells. It's akin to dispatching a highly efficient cleanup crew that specifically targets and removes the unwanted debris without harming the surrounding area. This technology has gained significant traction, particularly in treating Alzheimer's disease. Antibodies such as aducanumab have shown promise in clinical trials, where they effectively reduce amyloid beta plaques and slow cognitive decline, offering hope to millions. Another groundbreaking approach is enzyme therapy, which uses specific enzymes to break down the proteins in these aggregates directly. These enzymes act like molecular scissors, cutting the large complex protein molecules into smaller, non-toxic pieces that are easily cleared by the body's natural waste removal processes. For example, researchers are exploring the use of enzymes to target and degrade transthyretin, a protein involved in the common form of systemic amyloidosis. By breaking down the misfolded transthyretin before it aggregates, enzyme therapy can prevent the formation of harmful plaques in organs such as the heart and kidneys. The potential of these technologies to revolutionize the treatment of diseases associated with extracellular aggregates is enormous. Not only could they halt the progression of debilitating conditions, but they also offer the possibility of reversing damage by clearing existing plaques, restoring tissue function, and significantly improving the quality of life for affected individuals. Imagine a world where Alzheimer's or systemic amyloidosis are no longer feared as irreversible conditions, but are manageable through treatments that can literally clear the fog of disease. This vision is rapidly approaching reality, driven by the pioneering developments in immunotherapy and enzyme therapy. As these treatments advance and become more widely available, they promise to unlock new possibilities in medical science, turning the tide against some of the most challenging diseases of our time. The progress in these fields not only represents a leap forward in treating specific conditions, but also opens the door to applying similar strategies to a range of other diseases characterized by extracellular deposits. Welcome to the future of medicine, where clearing pathways in our body can restore health and vitality, offering hope where once there was none. Cellular senescence is like having workers in a factory who have stopped working but still hang around, taking up space and resources. In the context of our bodies, senescent cells are aged cells that have stopped dividing but refuse to die. They accumulate as we age, cluttering up our tissues, releasing harmful chemicals that cause inflammation and damaging surrounding healthy cells. This not only contributes to the aging process but also to the development of various age-related diseases, including arthritis, diabetes, and heart disease. The presence of senescent cells in our body disrupts tissue function and promotes chronic inflammation, leading to a cascade of detrimental effects on health. Importantly, these cells do not just sit quietly. They actively secrete a variety of proteins and inflammatory factors that damage nearby cells and tissue structures, a phenomenon known as the senescence-associated secretory phenotype, SASP. Imagine if we could selectively remove these problematic cells, like removing weeds from a garden to allow the remaining healthy plants to thrive. This is not science fiction. It's a scientific reality being addressed through innovative technologies such as senolytics and gene therapy. Senolytics are a class of drugs designed specifically to target and eliminate senescent cells. These drugs work by interfering with the protective mechanisms that keep senescent cells alive. For example, senescent cells often overexpress certain proteins that help them resist apoptosis, programmed cell death. Senolytics disrupt these pathways, effectively triggering the death of these cells. One exciting aspect of senolytics is their ability to selectively target senescent cells without affecting normal, healthy cells. This precision means treatments can potentially reduce the side effects typically associated with broader anti-aging approaches. Clinical trials are underway to explore the effectiveness of senolytics in treating conditions like idiopathic pulmonary fibrosis, chronic kidney disease, and even complications related to COVID-19. In addition to senolytics, researchers are exploring the use of gene therapy to combat cellular senescence. This approach involves modifying or regulating genes that control cell aging and senescence. By tweaking these genetic dials, scientists aim to either prevent cells from becoming senescent or restore their ability to enter the normal life cycle and die off naturally. Gene therapy provides a highly specific approach to potentially extend the health and functionality of tissues by ensuring that cells do not prematurely enter a senescent state, thereby maintaining a more youthful cellular environment. The development of therapies targeting cellular senescence represents a significant advance in our ability to not only treat, but potentially reverse aspects of aging. These technologies hold the promise of transforming our approach to age-related diseases, shifting from symptom management to actually addressing one of the root causes of biological aging. Imagine a future where treatments for aging are as routine as vaccinations, where conditions like arthritis and diabetes associated with aging are things of the past. Thanks to senolytics and gene therapy, this future is within our grasp. We are on the brink of a new era in medical science, one where we can potentially add not just years to life, but life to those years, enhancing the quality of life as we age. These technologies aren't just about living longer, they're about living better, with reduced chronic pain and increased vitality. Welcome to the dawn of a new age in healthcare, where cellular senescence can be managed or even reversed, heralding unprecedented improvements in health and longevity. Imagine your body as a complex network of fibers, gels, and fluids that form the scaffolding within and around your cells. This is the extracellular matrix, ECM. It provides structural support and determines how tissues like skin, blood vessels, and organs maintain their shape and function. However, as we age, this crucial matrix can become stiff due to the buildup of cross-links between proteins, primarily collagen and elastin. This stiffening not only reduces the elasticity of tissues but can also impair their function, contributing to conditions like arteriosclerosis, hardening of the arteries, skin aging, and impaired wound healing. When the ECM stiffens, it can lead to high blood pressure and reduced nutrient transport, making it harder for organs to function properly and for wounds to heal. Moreover, a stiffer extracellular matrix in the skin leads to wrinkles and decreased elasticity, visible hallmarks of aging. What if we could rejuvenate our bodies by restoring the youthful flexibility of the ECM? Advances in biomedical technology are bringing this possibility to life through innovative approaches like advanced glycation end product, age breakers, and tissue engineering. Age breakers are compounds designed to target and break the chemical bonds that form between proteins in the ECM, specifically those caused by a process called glycation, where sugar molecules bond inappropriately to proteins. These cross-links are a significant factor in the stiffening of the ECM. One of the most promising age breakers is a compound known as aligabrium, ALT-711. It has been shown in studies to break existing cross-links and significantly improve the elasticity of tissues, such as blood vessels and skin. This treatment could lead to better blood flow, lower blood pressure, improved skin texture, and enhanced overall organ function. Tissue engineering takes a more constructive approach by using biocompatible scaffolds that support the growth of new, healthy tissues. These scaffolds are designed to mimic the natural ECM and can be seeded with cells that will grow and replace damaged or aged tissues. Imagine a scaffold implanted in damaged heart tissue or within aging skin. Over time, the scaffold supports the formation of new, healthy tissue structures that integrate seamlessly with the body, restoring function and appearance. This method not only holds promise for anti-aging applications but also for regenerating tissues damaged by injury or disease. The development of age breakers and advances in tissue engineering represent a significant leap forward in our ability to treat and potentially reverse the effects of aging in the extracellular matrix. These technologies offer the prospect of turning back the clock on our tissues, making them more flexible and more functional. The promise of these technologies is not just in treating individual symptoms of aging, but in addressing one of its fundamental causes. By restoring the ECM to its more youthful state, we could significantly enhance our quality of life as we age, reducing the risk of chronic diseases associated with ECM stiffening and improving our overall vitality. Imagine a world where aging is no longer synonymous with a decline in physical capabilities or appearance. Thanks to the cutting-edge advancements in age breakers and tissue engineering, this world is on the horizon. These technologies are not just about extending life but enriching it, offering a future where each one of us can look forward to older years characterized by health, vigor, and resilience. Welcome to the new age of dynamic aging, where science meets vitality. Imagine if each cell in your body was a library filled with books, your DNA, that guide everything from your appearance to your susceptibility to diseases. But not every book is open at all times. Some are kept closed and only opened when needed. This selective opening and closing of books is orchestrated by a process known as epigenetics. As we age, however, this process can become disrupted, leading to the wrong books being opened at the wrong times, or some books staying closed when they should be open. This mismanagement can lead to a variety of age-related diseases, including cancer, Alzheimer's, and cardiovascular diseases. Dysregulated epigenetics involves changes in the mechanisms that regulate gene expression without altering the DNA sequence itself. These changes can affect how genes are turned on or off and can have profound effects on cellular function and health. For instance, genes that protect against cancer might become silenced through epigenetic changes, increasing cancer risk as one ages. But what if we could fine-tune this molecular script, restoring the youthful regulation of our genes? Emerging technologies in epigenetic therapy and gene editing are paving the way for such possibilities, offering revolutionary approaches to reset our genetic expressions to their optimal states. Epigenetic therapy involves drugs or other interventions that can specifically modify the epigenetic marks that regulate gene expression. These therapies can potentially reverse abnormal gene expression profiles by removing methyl groups, a common epigenetic modification that silences genes, using drugs that inhibit DNA methyltransferases, thereby reactivating beneficial genes, modifying histones, proteins around which DNA is wrapped, to change their influence on gene activity. Drugs like HDAC inhibitors can open up tightly packed chromatin to promote gene expression, or can tighten it to decrease gene expression. These therapies are not just theoretical. For example, certain HDAC inhibitors are already used in the treatment of cancer, where they reactivate tumor suppressor genes and help stop cancer progression. CRISPR, a tool famous for its gene editing capabilities, can also be adapted for precise epigenetic editing. This technique does not change the DNA sequence, but rather the epigenetic marks on the DNA. This can either increase or decrease the expression of specific genes. For instance, CRISPR and CRISPR, technologies derived from CRISPR-Cas9, where A stands for activation and I for interference. These tools can specifically activate or silence genes by targeting the epigenetic regulators directly, offering a highly precise method to correct dysregulated genes. The implications of these technologies are monumental. By manipulating epigenetic marks, we could potentially prevent or reverse age-related diseases, extend healthy lifespan, and improve our quality of life as we age. Imagine a future where epigenetic therapies are as common as vaccines, administered to correct epigenetic drift and maintain the youthful expression of our genome. As research progresses, the potential to harness these technologies becomes more tangible, holding promise for treating a wide array of conditions that currently lack effective interventions. We are on the brink of a new era in medical science, one where we can not only read the human genome but also rewrite the epigenetic errors that come with aging. Welcome to the age of epigenetic rejuvenation, where aging is not an inevitable fate but a reversible condition, and where the control over our genetic destiny is right at our fingertips. If you're looking for fresh, insightful content that keeps you engaged and informed, then hitting that subscribe button on our YouTube channel is a must. Not only will you stay updated with our latest videos, but you'll be joining a community passionate about exploring exciting topics and innovative ideas. But don't stop there. Liking our videos, commenting with your thoughts, and sharing our content helps us grow and means we can keep bringing you even more of the content you love. Each interaction not only supports the channel, but also increases our ability to create engaging, high-quality videos tailored to what you want to see. So, subscribe, hit the bell for notifications, like, comment, and share. Join us on this journey, be part of our community, and let's make this an incredible experience together. Your interaction makes a huge difference, and we can't wait to see you in the comments section.